Okay, today I'm going to talk to you about drug development. Now, in order to get new drugs onto the market, in order to treat people who are unwell, there needs to be lots of tests that happen to make sure that it's safe for people taking that drug. So what we're going to look at today is we're going to look at the different stages involved, what's the point of each of those stages, and also a little bit about blind and double-blind trials as well. So to begin with, in drug development, there's kind of two completely different sections. One is preclinical testing, and one is clinical testing. So preclinical testing just means before the drug is tested with people, and clinical testing involves testing drugs on people. So the very first stage of drug development is testing it in cells. So tissue culture in labs. So what you do is you get drugs and you put them onto different cells. So you could get some human cells here and you see other cells damaged by the drug. The cells responding how they should do when this drug is added. So at the moment, no one has been harmed. So on to the second step. If the first step, they've seen positive results in working on cells and tissue culture, then the second step, you test the drugs on animals. Now, a lot of people disagree with this stage. They think it's unfair because the animals can't give consent. They can't say it's okay. But the drugs are tested on animals. Again, the whole point of testing the drug on animals is to see if the drug is toxic and to what extent is it toxic, and to get an idea of dosage. So that's preclinical testing because we haven't tested on any people yet. The next test is to test it, number 3, on healthy people, or healthy volunteers even. So people volunteer to have the drug tested on them. So the reason why we test it on healthy people rather than people who are ill is, first of all, if they were ill, they might be weakened, and if there's any bad side effects, it might kill them or really hurt them. Secondly, if the person's ill, it would be difficult to tell if the side effects are caused by the drug or by their illness. So the point of testing in healthy volunteers is in order to find any side effects, and also to find the optimum dosage, the dose at the level of that drug where it works the best. Now, once you've tested it on healthy volunteers, our next step is then to test it on ill volunteers. So now we see if the drug actually works how it's supposed to. So here's someone ill in bed. Little thermometer. Right, so we then test it in ill volunteers in order to see if it actually works. Does it actually do what it's set out to do? Does it help this person who's ill with a certain disease? And the final one, once all of that stuff has gone through, remember this is clinical testing because it's with people. Once this has all gone through, the final thing that needs to happen is it needs to be approved by a medical agency. So that just means someone reviews, or rather an agency reviews all of this information that they've collected about the drug at every single step, and they say, right, is it safe to sell to the general public? And only once it's been approved, then we can sell to the general public. However, we need to know a little bit more about this stage here, about clinical trials. How do they work? So, clinical testing works a bit like this. So you have a doctor. So here's our doctor right here. And what they do is they have two different groups, okay? So you've got two different volunteers because basically the idea is you want to see if this drug works. So you're going to give the drug to one person, and the other, so one person is going to be given the drug, the other person is in a control group. So the point of a control group is to see if there's any difference between the drug group and the control group to make sure the drug's doing something. However, We can't just give the drug to the drug group and nothing to the control group because of this thing called the placebo effect. Now, what the placebo effect is, this person might just think they're getting better because they've taken something. It's like a psychosomatic effect. So what we do is we give this person in the control group a placebo. So we give them a pill that's just like this drug. It kind of tastes the same, it's sugary, the same size, everything like that, but only it doesn't have the active ingredient in. So placebo is like a dummy drug. Now, what we've got to do is we've got to make sure that the two people don't know which one is taking the drug and which one's taking the placebo. This is called a blind trial. So the patients don't know whether they're in the drug group or they're in the control group. Now, that's kind of one way. We actually go a step further because the doctor, if they know which group is which, they might be influenced subconsciously on their results as well. So what we actually have is something called a double-blind trial. Now, what this is neither the patient or the doctor know which group is the drug group and which group is the placebo group. So just to summarise, the different stages involved is preclinical, is before it gets to people. They test it in a lab on cells and tissue culture. They then, if it's safe, test it on animals, or if it's successful, they test it on animals to find the dosage and the toxicity. Then they move on to clinical trials. Clinical trials, remember, they can either be, well, ideally they're a double-blind trial, where neither the person taking the drug nor the control group knows which one's which. If it's successful, they test it on ill volunteers to see if it works, and finally gets improved by a medical agency, and we could sell it in order to try and help people around the world. Thank you.